All right, our next presenter is Carol, and she's going to be talking about Allen tools.
Hello. Let me get my Zoom screen set up so I can actually, well, I'm not sure that right now I can see you while I'm doing this. Usually I can view people, but I cannot right now. Okay, so bear with me. Thanks a lot for having me at the workshop today. I'm going to do a little bit of a different thing. Can you see my hand out on the window and my mouse moving?
Yes.
Great. I'm not gonna, I think Mike Horowitz this morning already gave a pretty good introduction to what we're doing at the BICCN and some of the tools we've been hosting. So I'm gonna start off by, I put the link to our handout in the chat. So you should be able to go through and click on links and follow along with some of the actions I'd like you to try as you go through. 27 minutes isn't a lot of time to go through a lot of things. I'm covering two things today. Our new Cell Types Knowledge Explorer, which is a place where we put together all of the information from the data about the primary motor cortex that came out of that package of papers that came out in October from the BICCN. We're going to walk through a little bit of what's there and then switch gears a little bit and walk through the BICCN data catalog and how to find data there. So if you see this here, I'm going to show first off how you start from this portal.brain-map.org. This is the Allen Institute's main portal for our data. From here, you can find menus to find your way to different types of data. There's a lot of data in here now after, I think, 17, 18 years of data generation at the Institute. And now we've been doing a lot more with these consortia and generating newer types of data. So what I want you to do first is scroll down here and click on New Way to Browse Data. So this is taking you to the data catalog. From the data catalog, you can either look at Allen Institute data, or you can go in and you can look at the BICCN data. But either way, I'm going to come back to this, and I'm going to take you first to the Cell Type Knowledge Explorer. This is one of our newest tools that we've put out, and it's not getting, hasn't been publicized quite as much yet. So we're going to walk through that here. If you go up to the top and click on Data Catalog, you get a drop-down menu. I want you to click on the Cell Type Knowledge Explorer. So this tool lives in the same site as our BICCN data catalog, and it currently serves to explore a multimodal cell census and atlas of the primary motor cortex. On this landing page, what you can see is we have taxonomies in here that were derived from those analyses from BICCN data for both for marmoset, human, and mouse. And so if you look over here on the left side, you can see how cell types are being aligned across the three species. There's a dendrogram across the top showing the relationship of these different cell types. And then you can see that in human, for instance, for the cell type right here, which is layer 2-3, IT, which should be intratelosymbolic projecting neurons. You might see, for instance, you have 6 cell types in the human, you have 4 in the marmoset that align to that category, and 3 in the mouse. So you can see this alignment across all of these. What I'd like you to see right now first is there's a little introduction to this page here. If you want to learn more about the data and the analyses, right-click on that flagship publication. You could open that up in a new tab and go straight to the Nature article that describes a lot of the analyses that pulled together information for these taxonomies. I should clarify that as part of the BICCN publications last year, there were a number of different taxonomies made. You can make a taxonomy based upon whatever data you have. You could make one just for mouse. And then if you're trying to bring data together to a cross-species framework, you might make certain adjustments to those taxonomies to adjust for that cross-species comparison. So this paper is your good resource for this cross-species analysis in this tool. So what this tool is trying to do is bring together all the information, the data, the summaries of how these data support different cell types. So I'm going to walk you through that a little bit. So I'd like you to go down and click on the mus musculus green button here. So each one of these gives you access to an individual species taxonomy. And so if you go down here to the mus musculus taxonomy, we've got this interesting rainbow colored representation of a taxonomy. It looks like a wheel, right? And what we have is in the interior are the highest level categories of cell types, the highest level classes. So you see GABAergic, non-neuronal, glutamatergic. In the middle layer of the wheel, we have some subclasses there that are fairly well recognized, but they represent kind of a large category of cell types. In here, you see nomenclature, a lot of it tied to neurotransmitters that we know are important in defining some of these different cell types. Like in GABAergic, we have SST-like, VIP-like, and P-valve-like. And if you go over the glutamatergic, they're defined more by the layer of the cortex that they're in, as well as what type of projection neurons these are. So if you see L6IT, that's a layer 6 localized cell type that has intratelocephalic projections. Around the outer part of the wheel is where we have the finer subdivisions of these cell types. And a lot of these are defined more by, this is all based on transcriptomic and epigenomic data. And so some of these will align to classically defined cell types, but many of them may look a little novel because these are cell types that are emerging on the basis of the great technologies we have now to interrogate transcriptomic expression in these cells. Excuse me. So if you go over to the left, there's some information over here about the taxonomy in general. Each taxonomy has an accession ID, and we have a Allen Institute community user post about how we've created those accession IDs. We use those. You can see it's in the URL as well up here. The data sets that were used to generate this taxonomy are all listed in the lower left. And if you click on the download button for any one of these, you can go to the NEMO data archive where you can go download the data associated with this taxonomy. And then if you click on the right, one of those green circles, you can actually get to NEMO Analytics and go explore the data at NEMO Analytics as you've just seen. Okay, so what I want to do to kind of go a little further into this is we are going to click on the P-valve-like cell type right here in the middle. And if you need that link, it's in the handout. And again, I can't see the chat or any faces, so someone please speak up if there's a question that I ought to pause for as I'm going along. Okay, so on this page is a summary of information about the P-valve-like cell types. This is, again, not a fine level cell type classification. It's a higher node in the taxonomy. So the information here is summarized across all the constituent cell subclasses. One main advantage of the Cell Type Knowledge Explorer right now over the other transcriptomics-based tools is that we are trying to integrate information from multiple sources and from multiple data modalities. So if you scroll down, you'll see we have summaries of metrics from not only transcriptomics, but we also have morphology, we have electrophysiology, spatial localization, and if it's available, we've got some epigenetics. Okay, so on the P-valve-like cell type over here, these types have formally been defined at this point in a provisional cell type ontology. And on the left side, you'll see the ontology, the accession ID for the taxonomy again, and you're going to see an ontology ID. What I want you to do, if you're following along, is right-click on that ontology ID, and it will take you to this cell type that's formally defined in the ontology. If you don't have a use for ontologies, that's great. There are a lot of people who are trying to link. This supports interoperability, that we have a formal definition of what some of these cell types are. So if you want to explore further, you can go look in here at in the OLS over here and see that the cell type shows up in several parts of the organization here of the ontology. It's defined by some of the markers it has. Over here, it says P-valve, the gene parvalbumin, and CNTNAP4 are two of the genes that are defining the cell type in mouse. And it's a type of GABAergic interneuron that's coming from the NGE, derived from the NGE. So there's a little more information here. If you're interested, you can go explore there and look at how the ontology is set up. At this point, I'm going to go back to our cell type Knowledge Explorer. And from here, want to show you what else you can do. Let's look at some of the plots. So in transcriptomics, right here, this is our gene expression plot. There's both a dot plot here and a UMAP over here. You can select which transcriptomic data set you want to look at. This one's defaulting to transcriptomics generated using the 10x V3 chemistry, using nuclei for single nuclei derived from mice. You can select other data sets if you want to change what gene expression you're looking at. We can make this window bigger by clicking on this double arrow in the upper right corner, and then you can look at these marker genes that were selected per cell type to help define what the cell type is. The selected cell type is highlighted over here in gray. So here's the p-value. You can see that we have 5,692, I think this is cells here. And you can see that this is really differentiated from its sibling cell types in the taxonomy by the high expression of p-value and CNTNAP4. There is another view if you want to explore a little bit more, which is this barcode view, which would show you a bit more about how these genes are expressed in here. I'm going to exit out of that. Okay. So if we continue on down and we take a look, there's too much in here for me to go through everything today, so I'm just giving you a quick preview. You can also look at morphology. So in the morphology view, again, we can expand this window. You can see cell type reconstructions that are currently oriented across layers of the motor cortex. So the pia is at the top and the white matter is at the bottom. You can see that this is showing exemplar images from the many cell type subclasses of this p-valve-like. So p-valve-like, as you recall, was in the middle of the taxonomy. There are many more refined cell types that you can access from there, and those are shown here. And you can see while some of them bear some similarity to each other, like these four all have soma localization in layer 5, you can see that some of the other p-valve expressing neurons have somewhat different localization and somewhat different morphologies. Okay, so I'm going to close out of that by clicking on the arrows up at the top. Feel free to go in and explore a little bit more in here, however you like. One thing I will point out is that if you click on the More Data button right here, you can actually go download, go to the project summary page. Can I right click it? Let's see. If I click that, it goes to the data catalog and it tells you about the data set that was used to generate this. In this case, it's a PatchSeq data set generated by Andreas Tolius over at Baylor. And you can find information about how to cite this paper here as well. Okay, where was I? Okay, so at this point, you might be wondering, how do you find the cell type you're interested in? Sometimes this nomenclature is a little bit harder for you to access. If you were looking at data from a historical perspective and you knew historical names, but you don't know what the genes are that are defining it in this particular taxonomy. What you can do is go up here to the upper left. Because of the great work our ontologists have done, including David Asumi Sutherland, Chris Mungle, and others at the Allen Institute, such as Patrick Ray and other contributors to the ontology work, a lot of the stuff has been pulled together so that we know, in some cases, which ones of these are aligning to some of those historical names. So if up here at the top in the search cell types box, if you can see this, I'll make the window a little bigger. You can start typing in a cell type you're interested in. In this case, start typing chandelier. And here you can see that the search ahead feature showed you 4 options right now. It's showing you 2 mouse options. There appears to be a division in the chandelier cells in mouse such that we have one category that looks transcriptomically distinct from a second category. But you also have options to go select and view the marmoset version of a chandelier cell or the Homo sapiens. I'm going to go ahead and select the mouse version of the chandelier cell. So in this taxonomy, our nomenclature here is p-val Viper 2_1, and that's because in this taxonomy, You should be able to see, where is PMAP in here? Oh, here's Viper 2. Huh, this one's, oh, here's Viper 2_2. Did I click the wrong one? I think I meant to click the other one, Viper 2_1. But in any case, you can also go through and explore the gene expression for this one. Okay, I was going to talk next for the next 10 minutes about searching the BICCN data catalog. Does anybody, oh, one thing I did want to point out, if you scroll down and you look at these more, the leaf nodes of the taxonomy, you can also use this graph at the bottom that's called the cross-species cell types. And this allows you to see, if you have a cell type, what the comparable cell types are in other species. In this case, for this particular one, there's not a great well, there's a high correspondence to a number of cell types in marmoset and human to this mouse cell type. I think I'm going to go up here because I think the other chandelier cell is a little more distinct. So if I did chandelier Viper 2 and I went down, you would actually see here that you can find this example of a chandelier cell has very high similarity to a cell type over here in human, and a very high similarity to a cell type over here in marmoset. Okay, really quickly, are there any questions before I move on to the data catalog? Okay, if there's no questions, I'll move on. So for the next part of the talk, I'm going to talk a little bit more about how to find data. The previous, in the Cell Types Knowledge Explorer, I had shown you how to go find the project summary page for some of these. That's part of the BICCN data catalog, and you can search for data for there as well. So I'm going to close these out. And if I go starting off to BICCN.org, the BICCN portal, this is something Mike showed this morning. This is a starting place for information about the consortium, how to access data, and how to explore the cell senses of the primary motor cortex. If you click, let's make this window a little bit bigger so you can see it. If you click on the data access tab up here, Mike showed this as well, you can see that there's different options. If you click this button, it will take you straight to the catalog, filtered for the BICCN data, or there are data download instructions for each of the data archives. NEMO, the Bill Imaging Archive, DANDY for electrophysiology and other physiology, and BOSS-DB, which is for EM and X-ray microtomography data sets. I'm just briefly going to also show you the Science tab. Again, Mike showed this morning. From the Science tab, we have a description of the primary motor cell senses of the primary motor cortex. So if you were to go all the way down, you can click on any one of these links and go look at the published paper. But we also have links out to where you can explore the data for each of these. So NEMO Analytics is hosting data sets from each of these, a lot of these papers. You can go look at the brain cell methylation viewer, CATLAS, cell by gene. The Allen Institute also has a transcriptomics explorer that I don't have time to show you today. And there's morphologies over at Neuromorpho. So I am going, from this diagram, you can also go directly to the data catalog filtered by these items. You can either go click for human, marmoset, or mouse, or look for data of a different modality. I'm going to go ahead and click the mouse image. And now this is taking us to the data catalog. From here, you're starting out just in a list of all the mouse projects that are part of the BICCN, 57 projects. If we want to filter this further, there's a filter button up here. Looks like 3 horizontal lines that are slider bars, and you can click it open. This is showing you the options by which you can filter, and it shows how much data is available in each option. One thing to note is if you select multiple items within one of these columns, it treats it as an or. But if you click filter options across different columns, it treats it as an and. So it's one important thing to keep in mind. I'm going to go ahead and add to it. Right now we have mouse and the Brain Initiative Cell Census Network are currently selected. And I'm going to add on to this, check the box for transcriptomics. To exit out of here, you can either click the filter button again, or here you can click in the window to exit that filter box. So at this point now, we are filtered for mouse transcriptomic data. We have 20 projects showing. I'm going to scroll down and find one of the data sets that was contributing to those taxonomies, which is this transcriptomic cell types in the mouse brain. And I'm going to open that. When you go to a project summary page, we're trying to give you all the information you need to be able to understand what is this data, how do you go get it, how do you cite it, under what license is it available. So from here, what you see at the top first is that you have links to the data collections. And so we have here a data collection of V3, 10x V3 data, and a collection of 10x V2 data. You might want to analyze them somewhat separately because they were generated by different techniques. If I clicked on one of those NEMO download buttons, it would take you to a landing page for that data collection, which you can then go download your data either by following the source data URL, which takes you to a directory style structure and you can download the data, or there are options for accessing data through the BD bag. So let's see, from here, if I go down, there's a project description. You can click on the more button to read more about this data. And you can see over here that this data is released under a CC BY 4 license. If you don't know what that is, you can click it open and read what that license is. There's also information on how to cite this data. On this particular one, there's a request to cite by publication, and there are 2 publications that are listed for the citation of this data. It was funded by this grant over here, and you could click that as well. And if you're interested in reading the protocols that were used to generate this data, you can also click on these protocols that are currently hosted at protocols.io. We've recommended to all of our data submitters that they publish their protocols officially through protocols.io or a similar site. Okay, so from here, this is where you found information about this project. Now, suppose you want to say, well, do I care about the data in this? Does it have stuff I want? Some of these projects, we are also adding explore links. So from here, you could go explore this data a little bit by going to the Transcriptomics Explorer, to NEMO Analytics, and the Cell Types Knowledge Explorer. So we'll hope to add some additional linkages in here so you can go view that data. What you can do right now is click on this Browse Specimens button over here on the right. This takes you to a page that has a listing of all the specimens. You can see in this page, these are libraries. That's a typical unit for our sequencing data of this is what was submitted, this is what was profiled. There's a library was generated and sent to the sequencing. This library from this 10x data was generated from a cell suspension. In this case, you can see the anatomical structure. It looks like some of these were dissected from MOS, motor cortex, secondary motor cortex, I think. And you can see what genotype of mouse that these came from. You can do similar things using a filter here to find cell types you want and filter down the list if you like. One thing that we've added is that if you click the download button up here, you can either go again to NEMO to download the raw data, or you can click on metadata or file manifest in order to get a zip file that gives you the specimen metadata here, along with a README file. Here, if I click this, it'll give you a README file that tells you all about the project. So it has all the information that was on that project summary page. And you can get the metadata for the specimens. And if I bring that over, well, my computer's not going to load that right away, but let's try it again. There we go. You can get the metadata for the specimens. And finally, what is useful is if you were to click the file manifest, you would actually also be able to get for each one of these. Okay. a link to the files that go to each of these specimens. So you have here's the specimen ID, which is your library ID. There's a file name, and you might have multiple files per specimen. And then for each of those files, if there was one you specifically wanted to go to, you could copy that URI in and go over to Nemo and pull up their landing page to see information about it, and then go directly to download that specific file. So when I clicked on that one, it starts downloading that tar file. I'm going to cancel that right now. Okay, so hopefully from this, you learned a little bit about how to search for data in our data catalog, as well as learned a little bit on how to use our Cell Types Knowledge Explorer to understand the summaries of what all these great transcriptomics data sets are telling us about cell types in the primary motor cortex of mouse, human, and marmoset. Thank you.